Assalamualaikum and hi everyone. Welcome back to SIL 1001. This is lecture 3 on the nucleus, intracellular membranes and also organelles. Right, in the last video you have watched you have learned about membranes, and today we're going to continue on the next structure in the cell, which is the nucleus. We're going to start off with nucleus. But before that, I'm going to introduce to you the endomembrane system. Well, see, inside the cell, you can find what we call the endomembrane system, which is a system of connected intracellular membranes that consists of the nuclear envelope, the rough ER, the smooth ER, Golgi apparatus, and also secretory vesicles and lysosomes. Anyway, this endomembrane system is different from, although the name here is membrane, is different from the plasma membrane that you have learned before. And this picture, this figure over here, describes the endomembrane system. The endomembrane system is important for the protein production, modification, and also transport. So we'll start from here. Nuclear envelope is connected to rough ER, which also continuous with the smooth ER here. You know that this area, this organelle over here, the rough ER, is important for protein modifications, especially for proteins that needs to be transported to certain locations, certain organelles, or to the outside of the cell, secreted from the cells. The membranes, after protein have been produced or modified in the ER, it will be transferred in a form of the vesicles. So the membranes and proteins produced by the ER flow in the form of transport vesicles to the Golgi. Okay, it will go to this Golgi, also referred as the post office. We're going to learn about this structure later on. And over here it will then be further processed and then will be targeted to their location. For example, some of the products of the proteins will be placed or will be in a form of the lysosomes, which later on will be available for fusion with another vesicle, for example, for digestions. And some of the vesicle carries protein to the plasma membrane for secretion, to be transported out, to be released to the outside of the plasma membrane. And the vesicles also important for the expansion of the plasma membrane. So the plasma membrane, the existing plasma membrane, can fuse with the vesicles, and then this will allow proteins to be secreted from the cell. All right, now moving on to the structure, the first structure that we're going to learn today, which is the nucleus. Nucleus is important or essential in eukaryotic cells that divide. It's the biggest organelle and controls indirectly all of the cellular activities. So nucleus is like the brain of the cell. The structure is spherical and varying in sizes. Smallest can be around 2 micrometer in fungi and then 30 micrometer in the dividing cells. So if you look at nucleus, you can see several components making the nucleus: the nuclear envelope, the membrane of the nucleus, the nucleolus, the chromatin, the genetic material of the cell, and then nucleoplasm. So it's like cytoplasm, but it's actually the fluid in the nucleus. Now, let's take a look at the structure. I mentioned before, there's the nuclear envelope. You can see nuclear envelope over here. And you notice that this nuclear envelope is actually connected to the rough ER. And then there's this, the membrane or nuclear envelope can be divided into the inner nuclear membrane, this color region over here, and also the outer nuclear membrane. And then other than this, you can also see the nucleolus, nucleolus here, and then nucleoplasm, surrounding, and you can also see chromatin, strands of chromatin inside of the nucleus. This is how the structure looks like under the microscope. So you see here is the nucleus. Yeah, you can see as this pars granulosa and pars fibrosa, fibril and also granules. Yeah, and then here is the nuclear organizing center. I'm going to talk about this later. Yeah, and then the nuclear pore on the nuclear envelope. Yeah, and then you can see that again. Yeah, you can see that the nuclear envelope is actually connected to the rough ER. So here you can actually see two types of chromatin, yeah, the euchromatin and also the heterochromatin. Yeah, I'm going to talk about, I'm going to elaborate a little bit more on the euchromatin and heterochromatin in the next few slides. Right, now let's, as I mentioned before, for nuclear envelope, there are actually two membranes, the outer and also inner membranes. Over here, yeah, you can see the outer and also the inner nuclear membrane, and perinuclear space for about 20 to 40 nanometer across. Yeah, let me see. Yeah, this perinuclear space over here, right? And on the nuclear envelopes, you can see nuclear pores connect the cytosol with the nucleoplasm inside the nucleus. The number of the pores vary in mammalian, typically with around 3,000 to 4,000 pores. And this pore is actually not just, you know, some hole, it's equipped with nuclear pore complex. Okay, the nuclear pore complex is like this. Okay, we'll see more about the nuclear pore complex later on. But coming back here, yeah, this nuclear pore actually control molecules that can go in and out, yeah, that can pass through the nuclear membrane or nuclear envelope. On the nuclear envelope, you can also see the nuclear lamina, you know, with lamin intermediate filaments support nuclear envelope and attachment of heterochromatin. If you see over here, a nuclear lamina would be here. It's actually kind of like part of the inner nuclear membrane, in purple, and the nuclear lamina is actually underneath it. It's very close to the nuclear. It's actually next to the nucleoplasm. So on the nuclear lamina, you can see lamin intermediate filaments, yes, kind of like supporting the nuclear envelope so that it won't collapse. This is how the nuclear pores looks like under the microscope, the freeze fracture micrograph. You can see those tiny bits over there, yeah, that represent the nuclear pore complex. The nuclear pore complex has intricate protein structure with 100 or more polypeptide subunits. Yeah, it is around 120 nanometer. Yeah, 120 nanometer in diameter. It has the wheel shape, yeah, octagonal. It has two parallel rings. Yeah, you see two rings over here, the blue color region of 8 subunits. 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 6 subunits with 8 spokes. Yeah, 8 spokes over here joining the ring. Let me see if I have another picture over here. Yeah, the 8 spokes over here in green color joining the ring. And also to the middle transporter. Yeah, this is the transporter. And the function of this nuclear complex, yeah, nuclear pore complex, is actually to transport material between the cytosol and also between cytosol and the nucleus, yeah, the nucleoplasm. So usually, yeah, what kind of proteins that will be transported will be, you know, ribosomal proteins for example. Yeah. Inactive mammalian cells, typically, sorry, not this one. Yeah. Around 20,000 ribosomal subunits synthesized per minute. Yeah, inactive mammalian cells, which means for 3,000 to 4,000 pores, around 5 to 6 subunits per minute per pore. Yeah. And another example, during replication, yeah, around 100 histomolecules per minute per pore are transported during the process of replication. This is a close-up view of the nuclear envelope showing the nuclear pores. Yeah, you can see those nuclear pore complex, yeah, on the membrane of the nucleus. Yeah. Okay, all right, I already explained about this. Same goes to this. Now we're gonna take a look at this structure over here. Yeah, the nucleus. Nucleus is the ribosome factory of cell. Okay, very important for you to remember this. Nucleus is the ribosome factory per cell. In a cell, you can find 1 or 2 nucleus, yeah, sometimes a lot more. You can identify the nucleus structure by the electron dense spherical region. Yeah, no membrane, it fills with fibrils and also granules. The fibrils are the DNA and also rRNA, and the granules are actually the newly formed ribosomal subunits. Yeah, you can see the dense spherical region over here representing the nucleus, yeah, granules and also fibrils. The nucleus structure is built, it's built by the nuclear organizing region, or in short, referred as NOR. In complete ribosome chromosome complements, yeah, there were always 6 chromosomes with a terminal nucleus organizing region, or NOR, over here. In most cases, yeah, part of the NOR was decondensed. So you see here, yeah, this part, this represent chromosome, yeah, and at the terminal, yeah, at the terminal of this chromosome, there's this NOR region, and these NOR regions usually are decondensed. Yeah? And several of these decondensed area will collectively form a big nucleus. In human, NOR contains genes. Yeah? Inside here, there's genes that encodes for 5.8S, 18S, 28S rRNA. Yeah, clustered on the short arm of the chromosome 13, 14, 15, 21, and 22. These are all the arcocentric chromosomes. Yeah, you're going to learn more about these rRNA are actually important to make the ribosome. Yeah, you're going to learn more about this in lecture 5. Here, let's move on to the ribosome assembly. Yeah, I mentioned before that the nucleus is important. It serves as the ribosome factory of the cell. Now let's take a look at how ribosome are assembled in the nucleus. Yeah. In the nucleus, yeah, the new ribosomal rRNA, yeah, over here. So it derives from the rDNA, yeah, always from DNA. So this part, the rDNA, ribosomal DNA, will be transcribed to produce the pre-RNA. So the pre-rRNA will then be further processed to produce the rRNA. Together, yeah, this rRNA combines with proteins, yeah, with ribosomal proteins imported from outside of the nucleus, yeah, coming in from the cytoplasm. So this ribosomal protein will come inside and participate in the ribosome assembly. So here, the ribosomal proteins together with the rRNA or RNA particle will then be used to make the subunit of ribosomes. So I hope we still recall, yeah, usually ribosome will be illustrated like this, yeah. There's the large subunit and also the small subunit, yeah. So this is to represent the small subunit, and this one is the large subunit. So you see here, the two subunits are not how do you say, fuse or bind together to form a complete ribosome structure. Instead, yeah, this large and also small subunit will be transported independently outside into the cytoplasm through the nucleopore, so that the ribosome can perform its function. Yeah? So the function of ribosome is actually to translate the mRNA into proteins. Okay. Right. Now we have learned about the nucleolus. Other than to produce ribosome, yeah, in the nucleolus that we've seen just now, the nucleus structure is also important to house or to store genetic material or DNA. So in the next few slides, we're going to look at the organization of DNA. First, let's take a look at the definition of genome. Yeah, when we talk about DNA in a cell, we need to understand the definition of genome. So genome is a complete copy of genetic information of an organism. So for eukaryotic cells, yeah, we have the nuclear genome, the mitochondrial genome, and also chloroplast genomes. Yeah, of course, in mammalian cells, you cannot find chloroplast genome. Yeah, so no, you don't say that all of the eukaryotic cells have this type of genomes. Yeah, no. So for chloroplast genomes, there are only like cells, for example, plant cells will have the chloroplast genomes. And the genome size increases with organism complexity. Usually, yeah, the prokaryotic DNA will supercoil and bind to small basic protein only. So they don't have nucleus, right? But for the eukaryotic DNA, they are a little bit more complex. So they will bind to histone to form these chromatin fibers and then further package to form chromosome. So we're going to learn more about this in the next few slides. But before that, let's do some calculation. In human, an average human cells will contain about roughly 6.4 billion base pair of DNA. And each base pair is 0.34 nanometer in length. So if you calculate, yeah, 6 billion, it's about 2 meters of DNA in each cell. But our cell is so small, the nucleus itself is just 10 micrometer. So how is this possible? How can you fit the 2 meters of DNA into this tiny structure of the nucleus? The secret lies in how the DNA is organized in the nucleus. Yeah, for this you need to understand the fundamental subunit of chromatin. Okay, we'll start from here. This is the basic structure of the DNA, right? Double helix. And this double helix is able, so I mentioned before here, DNA bind to histone. Okay, you see here, these beads over here represent the histone. Yeah? So the histone, it wraps around the histone structure. Yeah, and if we zoom in, yeah, we see this, yeah, how the DNA is wrapped around this nucleosome structure. And this nucleosome structure, yeah, this together, this the structure, the DNA wrapped around the nucleosome structure is called beads on a string. Yeah, it looks like beads on a string. This is under the microscope. You can see, yeah, this tiny shapes over here. They are the nucleosome, and the size of this is around 11 nanometer. So next, yeah, what happened next? Yeah, during the organization, this bead on a string later be further packaged into the 30 nanometer chromatin fiber. Yeah, additional with another histone protein, it will form the 30 nanometer fiber. The size is, yeah, by its name is 30 nanometer. This 30 nanometer fiber, yeah, will then be further packaged. Yeah, the next level of chromatin packaging is the folding. Yeah, you can imagine it's folded, folding of the 30 nanometer fiber into the what we call the loop domain. Yeah, loop domain, or the extended form. Yeah, or loop domain. Yeah. So the size of this loop domain will be in average around 300 nanometer. Yeah. This standard form of the chromosome or the loop domain will then be further condensed, yeah, to to the part of the chromosome structure over here. Yeah, you can see here, further condensed into the heterochromatin structure. Yeah, this is how it looks like under the microscope. Yeah, this segment of chromatin is so highly compacted that they appear as dark region under the micrograph. Yeah, it's called the heterochromatin. I think yeah, we have seen those before. Yeah, this is the heterochromatin. Yeah, appear as a dark region because they are highly condensed structure. And usually, yeah, this heterochromatin and also the euchromatin that I mentioned before, they are associated with the activity of the DNA. Okay, whether they are active or not. So I'm gonna go back. I'm gonna come back to this slide, but let's continue on the organization of the DNA, and then later, yeah, it will then be further condensed and duplicated. For example, in the dividing cells, yeah, they will be duplicated, replicated, and duplicated, so that you can see this chromatin structure, yeah, like this. So that's the organization of DNA and how it is organized in the nucleus. Yeah. So for your information, yeah, the DNA won't appear always like this in the nucleus. Yeah, you can only see the structure over here during the mitotic stage of the cell. So interphase, they would appear differently. Yeah, I'm going to learn about that in lecture 6. All right. Anyway, let's come back to this image over here about the nucleosome structure. Yeah, or this one, the bits on a string structure. Okay, if you look closer, this region over here, this structure complex over here, it is the nucleosome. Okay, I misspell it. Nucleosome. Okay, and the nucleosomes are made of different subunits of the histone protein. Yeah, you can see H2 of H2A, 2 of H2B, 2H3, and 2H4. Okay, so DNA, yeah, around 146 nucleotide pairs will wrap around one nucleosome structure or nucleosome beads. Yeah, there are 8 histone molecules inside one nucleosome structure. Yeah, as I mentioned over before, yeah, H2A, H2B, H3, and H4. There's another histone protein, yeah, not part of the nucleosome, yeah, which is H1. Okay, so H1 serves as a linker, yeah, as a linker in order to form the 30 nanometer fiber. So without the H1 subunit, this nucleosome on a bead, beads on a string, won't be able to form the 30 nanometer chromatin fiber. How the DNA can wrap around the histone protein due to the to the charge. Yeah. For your information, DNA is negatively charged. Yeah, but the protein, yeah, the histone proteins, they are positively charged, yeah, due to the content of lysine and arginine, yeah, amino acid. So different amino acid would have a different polarity, yeah. This will, give rise to different charge of the protein. So positive, negative, there will be, they are able to bind together. So this one I already mentioned, yeah, using this figure before, so we can skip that for now. Now, let's take a look on the packing ratio. Okay, packing ratio is just the meaning of packing ratio is total extended length of the DNA molecule divided with the length of chromatin or chromosome. This is, to see how can the 2 meters of DNA be placed inside the 10 micrometer nucleus. Yeah. So basically, bit on a string, bit on a string, the packing ratio is 7. Okay, well, how you got this? Yeah, I'm gonna show this to you. Okay, for just for an example, the shortest human chromosome contain around 4.6 times 10 to the power of 7 billion base pair of DNA. Yeah, and it's equivalent to the size of 14,000 micrometer. But anyway, so you know that the size of one base pair would be 0.34. So we times 0.34 nanometer, yeah, which will give you around 1564000 nanometer, yeah, in length. And this is also equivalent to 15640 micrometer. Yeah, so how this yeah, the size of the and we divide this with 2. Yeah, divide it with 2 micrometer. So why 2 micrometer is because the chromosome size would be around 2 micrometer in length. Okay, chromosome size, so which will give you around 7.8, roughly 7 packing ratio. Okay, and the next level would be the formation of the 30 nanometer fiber. So the 30 nanometer fiber would have around 6 bits per turn, and it will give rise to the packing ratio of 42. After the 300 nanometer fiber formation, yeah, this would be about how to say 750 packing ratio. It is 15 to 20 times more compact to the 30 nanometer fiber. Yeah, and then finally, yeah, the formation of the chromosome structure itself, which will have around 8,000 to 20,000 packing ratio. Don't worry so much about all of the maths and all. Yeah, if you couldn't understand very, very clearly, it's just to show you the compaction relative, yeah, to the previous structure. Like for example, this bits on a string, it is when you compare, if let's say you compare just DNA, the packing ratio is just 1 at that time, 1 time. So bits on a string, it will be 7 times more compact compared to the DNA without any formation of chromatin, without any binding with the histone protein. And then here it's 42 times more compact compared to the bits on a string structure, yeah, so on and so forth. So at the end, that's why the 2 meters of DNA is able to be packed into the tiny nucleus structure. Other than the histone protein, just to let you know that there are other proteins, yeah, other non-histone proteins with a variety of enzymatic, structural, and regulatory roles, yeah, important for the chromatin structure. Okay, let's come back to this slide. Yeah, I've previously mentioned that if you look under the microscope, you are able to see two types of chromatin, yeah, the euchromatin and heterochromatin. In the organization that we've seen before, we see the heterochromatin structure, highly dense, but not all region, yeah, will be like this all the time, yeah. You can see this structure in the mitotic division stage, yeah, but in other stage, yeah, for example, in the interphase, yeah, some of the chromosome of the chromatin will appear as euchromatin, yeah, and it depends on how they are packed, and or whether they are loosely packed or they are tightly packed um, with the help of the nucleosome structure. For this, yeah, you can imagine that the chromosome or the chromatin are loosely packed, yeah, in a diffuse form of the DNA. This usually associated with the actively transcribed genes, yeah. And opposite to this, the heterochromatin usually associated with transcriptionally inactive genes. Yeah, if they are like densely packed, then you know that for transcription, there we require enzyme, for example, the RNA polymerase. So if they're tightly packed, RNA polymerase cannot go to the DNA and then transcribe. So that cannot happen. So this usually, the structure over here usually associated with the transcriptionally inactive form of the DNA and appear as dark spot under the electron microscope. Yeah. So during division, our chromatin becomes highly compact. Yeah, so that's why we can see the metaphase chromosomes. All right. Okay, next, the nucleoplasm is the nuclear matrix, which contains nuclear proteins, enzyme, metabolites for RNA and DNA synthesis. Yeah. It communicates with cytosol through the nuclear pore. Right, function of the nucleus: store genes on the chromosome, organize genes into chromosome to allow cell division, transport regulatory factors in gene products via nuclear pores, produce mRNA that code for the protein, produce ribosome in the nucleolus. Yeah, you have seen this before. Yeah, organize the uncoiling of DNA to replicate key genes. Okay, so these are the function of nucleus. Details of DNA and RNA will be dealt with in the section of molecular genetics. So you're going to learn about, the DNA, the transcription, translation, and all with Professor Zulkarnain. Okay, so that's the end of the first part of lecture 3. So next video, I'm going to continue on the intracellular membranes and organelle.